All right, I think we will get started here. So hello and welcome everyone to our fourth Inscopix Journal Club. My name is Jonathan Nassi and I'm the Director of Translational Science here at Inscopix. We began this series a few weeks ago to help keep the scientific community connected during these difficult times. As part of this series, we'll be discussing publications of importance to the field of circuit neuroscience, often featuring Inscopix and Vista or Invoke imaging. And we're excited to have you join the discussion from your virtual offices at home. You can send us questions via the chat window throughout the presentation and for up to 5 minutes after the presentation is over. We'll do our best to get to most or all of the questions with a hard stop right at 12 p.m. Pacific time. Today, our field scientific consultant, Kyesang Chen, will be presenting a Nature Communications publication from Larry Zweifel's group at the University of Washington. Kyesang received his PhD at UC Berkeley in Yang Dan's lab and has extensive experience applying in vivo calcium imaging techniques, including Inscopix miniscopes, to study the hypothalamus and deep brain regions during sleep states. And so with that, I will hand it over to Kyesang. Thank you.
Yeah, thank you, Jojo, for the introduction and your participation for this webinar. So I'm going to discuss this paper with you, and the title of this paper is Persistent Activation of Central Amygdala CRF Neurons Help Drives the Immediate Fear Extinction Deficit. So in a classic Pavlovian fear conditioning, people usually put different stimulus together, the unconditioned stimulus with the conditioned stimulus, CS and US. So usually following the US, there's a corresponding behavioral responses. So people tie these two different stimuluses together. So then for a conditioned stimulus will induce the nature physiological change together. So as you can see on the right side for this example, they put CS with the specific tones to the animal, which is a rodent here, and US is electricity full shock. So when they pair these two things together, the CS-US chance putting together, and after the US, usually there's a freezing. And when they put these two things together, the chance for freezing percentage getting higher and higher and higher. And after this training period, even though when the rodent were presented with the CS, the tone alone, the animal still demonstrated freezing behavior. That's a classic Pavlovian fear conditioning. And there's another thing we are going to talk about today is the fear extinction. So after the fear memory was formed, if you try to present the animal with the CS alone without US, basically there's no full shock to the animal, the percentage of the freezing behavior will gradually go down till almost to the baseline. And you can try to test the efficiency for this fear memory extinction by doing the CS alone probably a day later with just a single tone to test what's the percentage for this animal to show fear extinction. So in this case, you'll be able to test the fear extinction efficiency. So in the earlier research, people have already found the extinction training that occur proximal to the conditioning is less effective than the extinction training that follows a delay. And the mechanism behind it was proposed that the immediate fear extinction deficit is the result of an active fear state and associated stress arousal system linked to the net recency of fear, then antagonize the extinction process. And regarding the neuromodulatory system involved in this process, there were two major neuromodulatory factors that were believed are important for this process, which are the non-adrenergic neurons, this system, and also the corticotrophin releasing factor, the CRF systems. These two systems were believed in the modulation of these immediate extinction deficit. And regarding the neural circuits, people have found many different nodes in the circuits that are important for this process. And the central amygdala, CEA, has emerged as a critical site in the early formation of the fear memory. And there were a certain percentage of these neurons in the CEA are actually express the neuropeptide CRF that we mentioned earlier, which are important for the fear memory regulation. Also, the CEA CRF neurons that actually has the ability to regulate many different fear memory associated nodes, especially the noradrenergic neurons in the LC. So taken together for all the previous research results, the CEA CRF neurons were believed to be an important component for the neural circuit associated with immediate extinction deficit associated with the residency of fear. And in this paper, the authors try to understand and examine the role of these CEA-CRF neurons, especially for their role in immediate extinction deficit associated with the residency of fear. So since we talk about the memory extinction, the fear extinction experiment, so here they try to compare two different behavioral diagrams. So in the first one, called immediate extinction, you can see here is very similar to what we saw in the earlier example. There's an acquisition, which is a CS for 9.5 seconds, and there's a 0.5 second US push shock followed right after the CS. And they will repeat this protocol for 5 times. And after 20 minutes, they will perform the extinction protocol, which is CS alone for 10 seconds, and they repeat it 30 times. And 1 day later, they would do the recall experiment by presenting CS 10 seconds CS for 4 times to the animal. And the other protocol is a delay extinction. Basically, it's almost the same as the immediate extinction. The only difference is the time between acquisition and extinction were extended to 24 hours. Basically, 1 day later, they will do the extinction. That's a major difference between these two protocols. And to really examine the role of these CEA-CRF neurons during this process, they express these TTX toxin to block the synaptic transmission in the CEA-CRF neurons. So they took a CRF Cre mice and injected these Cre-dependent TTX to block the activity of these neurons. And you can see here, because they also label these neurons with GFP, you can see the green color in the CEA-CRF neurons. So here they try to divide the animal to 5 groups, and each group they have 12 mice. So the first group is the negative control with no extinction at all. And the first pair is a TTX and a GFP for the immediate extinction experiment. And there's the other pair is a GFP and TTX with delay extinction experiment. So basically during the acquisition or the extinction, you can see the freezing behavior getting higher during acquisition, and during extinction is gradually going down. But there's no significant difference among these groups during these 2 periods. However, during the recall period, we can see here, with no extinction, when I try to recall the memory by presenting CS alone to the animal, there's a 60% freezing behavior that will observe. And during the GFP immediate group, they express a GFP to animal, but there's nothing blocked. And for the immediate extinction protocol, the percentage was comparable to the no extinction group. However, when they try to block the activity of these neuron during in the CEA-CRF neurons, and in the immediate extinction group, the freezing percentage were significantly lower compared to the control groups, which means that the activity of these neurons are actually antagonize immediate fear extinction. And for the delay group, they were still able to observe the decrease as well. So since so far they found the activity of these neurons are important for immediate fear extinction deficit, they also want to examine the neuropeptide CRF alone. Especially for the people who are studying neural circuits, people always wonder whether this kind of modulation were done by those classic neurotransmitters or the neuropeptide itself. Especially in this case, the CRF peptide were also proposed to be important for this immediate fear extinction deficit. So here they took a CRF Cre mice, and in this time they try to express this vector into the animal to really do the homologous knockout. So as you can see here, on the right side is the heterozygote, and on the left side is the homozygote. And here they try to stain the Cre with a GFP. So these cells are the CRF neurons. And on the right side, the heterozygote, they can stain a CRF peptide, but on the left side, there's no peptide stain at all. suggested that they were able to knock out the CRF peptide completely. And on the right side is the behavior diagram that we just took a look. Here is the immediate extension protocol. Again, there's no significant difference during the acquisition nor in extinction. However, during the recall period, you can see the percentage of CRF knockout group were actually lower than the control group, suggesting that these CRF neuronal peptides are important for these immediate extinction deficit. So far they found like the activity and the peptide itself are antagonizing the fear extinction. So they want to do a specific manipulation of this neuron activity to see whether the activity of them are really necessary during the fear memory extinction. So here they express DREAD into the CEA-CRF neuron. They use a cree-dependent DREAD HM4DI to label these neurons. And then they try to inject the CNO right before the extinction period. So as you can see here, when they try to suppress the activity of these neurons, there is no significant difference during the extinction. But again, at the recall period, they found the percentage of these animal. The freezing percentage was actually lower than control, which again support the idea like the activity of these neurons are actually antagonizing the fear extinction. And during the time window of the extinction were especially important. And they also did a complementary experiment by stimulating these neurons using HM3DQ DREAD. So this time is different. They are using the delay extinction protocol. So usually during the delay extinction, the animal were able to perform better for the fear extinction. And again, they injected the CNO particularly right before the extinction starting. So there were no significant difference in the first two part of the experiment. However, during the recall period, you can see here, when they try to inject the CNO, the freezing percentage were higher than the control, suggesting that the fear extinction wasn't as good as the control group. So far by these three experiments together, we know that the CEA-CRF neurons activity are actually antagonizing the immediate fear extinction by TTX as well as by the DREAD. And the CRF peptide itself is also important for this process. And particularly based on the CEA-CRF neuron experiments with the DREAD by manipulating their activity, either during the delay period or immediate period, we can try to have a hypothesis that the CEA-CRF neurons activity are higher, should be higher during the immediate extinction than the delay extinction period. But to really answer this hypothesis to examine whether it's true or not, we need to check the activity of these neurons. Therefore, the author decided to use Inscopix NVISTA system to check the activity of these CEA-CRF neurons. So basically, they use CRF pre-mice and express cree-dependent GCAM6M into these cells, as you can see here. And they implanted the green lens and also the NVISTA miniaturized microscope and performed the CSUS experiment as we just saw so far. So here are their results. So in the panel D, here is a single cell example during the all 4 periods of the experiments. So before the acquisition and extinction, the add-1 habituation period, basically they will present 5 CS alone to the animal without any US, just a tone alone to stimulate the animal to see what the response is. And there will be 5 CS US pair during the acquisition, and then will be either 20 minutes later or 1 day later, they will do the extinction protocol for 10 times, and they want to observe what's the activity change. And another day later, they will do the recall experiment to test the activity of these neurons. And on the right side is the population average of these cells. So as you can see here, for this habituation part, in the beginning, there was some activity showing accompanied with the CS, but during the habituation, this activity were actually erased to similar to the baseline. And during the acquisition, you can see at the beginning for both immediate and the delay group, the activity were actually highest right after the CS, which is the US part when the animal were provided with electrical full shock. However, during the CS-US pairing to the animal, you can see the activity gradually shift to the beginning of CS. That's true for both cases. What's really interesting is in the next page. So you can see here, here's the extinction protocol during this period. And for the immediate extinction group, you can see the average activity of these neurons were highest during in the beginning of the CS, and less consistent across the 10 times of extinction protocol. However, if you check the delay extinction group, you see the activity of these neurons were highest in the beginning, but actually dropped very quickly till very close to the baseline in the end of this extinction protocol applied to the animal. So here is a quantification data. You can see the slope for the immediate extinction group were pretty flat. There was no much change across the 10 times of extinction protocol application. However, you can see a very dramatic change of the slope for the delay extinction group, suggesting that the neural activity changed very quickly during these 10 times of extinction protocol applied to the animals. And 1 day later, they performed a recall experiment to the animal. And this time, the activity of this immediate extinction group, this neural activity was higher than the delay extinction group. And it was true in the beginning until the end of the recall experiment. So it seems like there are some significant difference between part of the whole protocol they apply to the animal. So they also want to take a more systematic manner to really examine whether it's true or not. So they decided to do a decoding accuracy test. So basically, they try to find the calcium trace during the CS between the immediate and the delayed extinction group, and then perform the PC analysis to find the major component that contribute to this calcium trace shape. And then they use this naive Bayes Gaussian classifier to distinguish between the immediate group versus the pool data from everything altogether, and also for the delay group. And then they did it for the bootstrap for 4,000 times and to see how likely the algorithm were able to distinguish the immediate versus the delay group. And they run it for all 4 periods like we just looked at earlier. And the algorithm was only able to distinguish the extinction and the recall part, suggesting that these 2 periods are the major differences between the activity of this neuron during the whole fear extinction protocol. So to really take more careful and systematic look of these neurons, the author decided to check the neurons that they were able to identify across the whole fear extinction protocol. Basically, they are particularly looking at the animal, the cells they were able to find from the beginning of habituation through acquisition, through extinction till recall period. So for the cells, they were only identified once or twice or 3rd time in the process, they won't look at it. So on the right side were actually the percentage of the cell they were actually look at in the end in this following figure. So 1 times 100%. And here you can see when you try to align the cell from trial to trial, the percentage becomes lower and lower when you perform the longitudinal registration. And till the 4th time, they were able to register around 15% of their cells. So here's a result. They found 25 cells were registered across the 4 sessions and for the immediate extinction group, and 23 cells for delay extinction group. So basically, there's no much differences for the habituation part. And during acquisition part, you will see the activity of these neurons in both cases were very similar to what we saw earlier. They were highest during the US part, but shift to CS in the beginning of CS become their peak in the end of the acquisition. But amplitude here seems to be higher compared to the pool data that we saw earlier. And it's also true for the extinction part. The activity was actually higher in the beginning, but dropped to the baseline till the end for the delay group. And for the immediate extinction group, the activity still remained reasonably high till the end. And here's a quantification data showing significant differences between these two groups. And the recall part, when they did this registration, it became even clearer. The activity of these immediate extinction groups still remain quite high till the end, but there's almost no change for the recall for the delay extinction group. And it also shows significant differences for the quantification data here. So by doing the in vivo calcium imaging using the NVISA system, so far they were able to know the persistence of the CS evoked increase in calcium signal during, especially high during immediate extinction and also the extinction recall. And this suggests like the activation is critical for the observed necessity of CEA CRF neurons in driving the immediate extinction deficit. So to really test this idea, to really know whether the activity of these neurons are really important during the CS, and this part involved in the fear extinction deficit, they decided to shut down the activity of these neurons specifically during the CS part during the extinction. So basically, they use a JAWS, which is an inhibitory opsin. They express JAWS into the CEA CRF neurons, and they use the laser to turn off the activity of these neurons. And here's the immediate extinction protocol. Everything was the same as what we saw earlier, except for this part. During the CS part during the extinction protocol, basically only 10 seconds for 30 times, they turn on the laser to shut down the activity of these neurons. So as you can see here, there's no significant difference during the early acquisition nor extinction part. But when they turn off the activity of this neuron during extinction part, and then they recall the field memory, the freezing percentage was actually lower compared to the GFE control, which means that the activity of these neurons are actually antagonizing fear memory extinction, and the activity of this neuron during the CS in the extinction part are really essential. So far, all the experiments are focusing on fear memory extinction. In the end, the author decided to do one opposite experiment. It's called the fear reinstatement. So by the earlier research results, people found the following extinction of a CR to a CS. The CR can be reinstated by presentation of an unpaired US. And also other research show that antagonism of centrally active CRF is also sufficient to prevent US-induced reinstatement. So in the last experiment of this paper, the author wants to test whether the activation of CACF neurons following extinction training is really sufficient to reinstate a CR to the CS or not. So to do so, they used a channelrhodopsin to activate these neurons optogenetically. And on the right side, you can see the protocol here. It's very similar. It's acquisition, extinction, and recall. But in between extinction and recall, they add one extra session called reinstatement. This reinstatement was performed 1 day after the extinction, and it was done for the delay extinction group. So there are two protocols they were using. The first one is a classic electrical full shock, or they did optogenetic activation to specifically manipulate the activity of these CEA-CF neurons. So again, there's no significant difference for the earlier part. But what's really interesting is here is in the end, during the recall period, you can see the control group, the freezing percentage was around 25%. However, when they add one reinstatement protocol for the full shock with a full shock, the percentage goes up to around 50%. And these results were able to be mimicked by activation of the CEA-CF neurons, suggesting that these neurons' activity were actually sufficient to induce a fear memory reinstatement. So taken together for what we have seen in this paper, so by in vivo calcium imaging, especially, the author found a persistent activation of CEA-CF neurons during immediate extinction training and also the memory recall period. And the activity of these neurons were actually higher compared to during the delayed extinction training period. And they used a series of optogenetic and also chemogenetic activation or inhibition. They found the activity of these neurons are actually important for this memory extinction process. And also the neuropeptide CRF itself is also a contributor for this process. It's not only the neural activity, but also the peptide is important. And lastly, by the last experiment that we just looked at, they found the activity of these neurons were actually able to reinstate the previous extinguished fear responses. So taken together, these results are consistent with the CEA-CF neurons being an important contributor to the stress arousal system associated with the residency of fear that has been proposed to underlie the immediate extinction deficit. So basically, that's the this paper. They use the optogenetic, chemogenetic, as well as the in vivo calcium imaging using Inscopix NVISA system to examine the activity of these neurons and to manipulate them during specific time window of the fear extinction protocol to really examine the correlation of their activity with freezing behavior and the causality of their activity to the fear memory extinction. Yeah. So thank you. That's the content of this paper. I'll be happy to take questions.
Great, thanks, Kyesang. That was excellent, really clear. Thanks for taking us through the paper. And yeah, I really liked. I thought it was really interesting to see the calcium imaging data and see that maintained activity level during the immediate fear extinction as compared to the delay, where you see the activity over time reducing. I thought that was actually pretty striking kind of finding with the calcium imaging data. Yeah, we do have some questions, and of course, people are free to continue to submit questions as we go along. One was more of a comment, I guess, to some degree, but I guess curious if you have any insight into this. In figure 2, where they do the conditional inactivation of the CRF encoding gene itself, that's actually an approach that you don't necessarily see as often in a lot of these papers. You know, maybe you could just explain a little bit more the approach here, because it looks like to me they're actually injecting, in this case, something that's going to express Cree, and then they're using 2 different lines of mice that basically have this gene flanked by LOXP sites in one case, in the homozygous versus the heterozygous. Yeah, I guess the comment or question is really just more, you know, we're familiar with a lot of Cree lines that are out there for targeting specific cell types, for instance, but curious how many of these types of lines are out there, if you happen to know of others, or are familiar with this sort of approach to actually, you know, essentially inactivating a gene for a particular neuropeptide in this case.
Oh, I see. Yeah. So let me try to make this approach clearer. So it was actually a Cree injection. here actually, they injected AAV-Cre-GFP into this region. And particularly, it was actually a mice that has the LuxP site that needs the Cre to drive the homologous recombination to really knock out the exons in the CF genes here. So you can see here, basically try to take out one of the exons for the whole gene so that the peptide won't be able to synthesize. So this was actually, it's not like common these days for the neural circuit mapping paper, either in Inscopix or other paper they were using optogenetics or DREAD. This is not like common these days, but this was an approach that has been widely used in the synthetic protein transmission. For example, I think Sudolph lab in Stanford, they have been widely generating tons of these mice or knocking out different neuronal transmitter, and I mean the transmitter, the receptor or the synthetic proteins, and try to examine these the system. So this was actually a little bit, you can say it's a traditional or classic method that have been widely used in the earlier literature. It's just less common these days for the people who are studying the neurons using those more cutting edge technology. But this method were actually providing a great insights overall for examining the roles of these cells. And recently, people have also adapted this methodology by using CRISPR Cas9 system. They were able to replicate these method by just injecting Cas9 to a particular brain region. So in that case, it's much easier. You don't need to generate any Cre mice anymore. You can just basically use a Cas9 mice with a CRISPR to knock out the gene wherever you want. That's probably much easier. Yeah.
No, that makes sense. But yeah, I think it's definitely interesting to be able to move beyond, in some cases, just manipulating the ultimate output activity of a certain population of cells and actually try to probe specific genes and signaling pathways that population might be involved with. So I thought that was a nice addition here.
Yeah.
Great. And then if we go to figure 3, I think there was just a question about the fact that it looks like in those experiments, they were using dreads. And in one case, they inhibited, I believe, during immediate fear extinction. And then in the other case, they activated during the delay fear extinction. And I guess just one comment that came in, like, would basically doing the opposite, would activating during the immediate fear extinction, kind of have the even worse extinction recall later? Would it, would the prediction essentially be the opposite in that case? I guess those are experiments specifically that they didn't actually run, but is sort of the reverse experiment. Is that the expectation that we would expect?
Yeah. I think that depends on how low is the activity of these neurons, because I think based on their hypothesis across the whole paper, they believe the activity of these neurons were actually higher during the immediate extinction part. So in this case, if you try to turn on it even more or try to suppress, even more during the immediate extinction, or try to suppress it even more during the delay period, it may be very close to the baseline or the maximum ability for these neurons. So they may not see a clear change for the freezing percentage. Yeah, that can be the case, but they didn't show anything. So that was just a possible explanation of the scenario. Yeah.
Right. Yep. Yeah. And one other comment was just an observation that you may have also found but in some of the extinction plots, the data certainly doesn't, it looks non-overlapping in some cases. I think they do address this in the discussion. I think in most cases they say that there wasn't a significant effect during the extinction. And I believe in the discussion, they may have done some sort of group analysis across their experiments to try to back that up further. But I don't know if you had any further insight or comments on some of those plots. Yeah, like the one you're showing.
Yeah, like this one, right? Like in the end, it still looks not overlapping, right? So I would say it's about statistics, how you do it, right? If your n number is high enough, you will still be able to get a significant difference in the end. It's
Right.
about the behavioral or the animal number they design for a particular experiment. But you can say this one is easier to reach significance, maybe more obvious to see, but it's not, I mean, obviously there are still some differences here. It just the way it was not able to prove to be significant based on the animal size they are using in their behavior diagram. Yeah.
Yeah, makes sense. Yep.
Yeah.
Yeah, I mean, some of the trends at least are sort of in the direction that that you might expect if there were to be some effect.
I believe there. I mean, it shouldn't be the simple like only involved in the recall something. It's basically disturbing the whole memory extinction protocol. Yeah,
Right.
I think that's my understanding.
Right.
Yeah.
Right. Yeah. Another question that came in that I think is relevant to figure, let's see, maybe figure 5, where they're doing longitudinal analysis, is just asking whether the decrease in number of cells collected with a longitudinal registration across behavioral sessions might be due to some sort of pruning of the neuronal ensemble as the animal goes through and learns these different phases of the behavior, or is it because of another biological reason? My sense is that if they're referring here to the sort of across number of sessions, the proportion of neurons, they do cite some other work in the BLA looking at how many cells kind of are active across different phases. In my mind, I would have expected, I'm not sure we can pull this out of panel sub the A panel, but I would think that there'd be pretty good overlap between, for instance, like the habituation and acquisition phases, which did those happen on different days? Was that like, or was that just prior to acquisition they have habituated for?
The first two should be the same day based on my understanding. Yeah.
Okay. Yeah.
Yeah. So it looks very similar actually for most of the cells. Yeah.
Right.
Yeah.
Yeah, I don't know if you have any other thoughts about this. I mean, certainly in each of these phases, there's different things going on. And so certainly it's not surprising that you're going to have some cells, for instance, during acquisition where you have the US occurring, where you're going to have cells that you might not have active, during habituation or even, extinction or recall. So to some degree, some of this lack of, kind of the reduction in number of cells across session makes sense, probably just based on what's happening during these different behavioral phases and the cells that are expected to be active during those.
Yeah, I think they do make sense, right? Because it may not be like one single cell must be involved in extinction or recall. It's not really a necessary thing. Right. But I think when they try to screen the cells here, they particularly focus on the neuron that responds to both CS and US during the acquisition period. That was the criteria they selected for the cells. So there's a high chance that you are actually seeing the cells that may respond to CS and US because CS and US are actually presented in all 4. I mean, especially CS are represented during all 4 sections. So it should be expected to see the same cells across the 4 sections. Yeah.
Got it. Yep. Excellent. Let's see. One question I had actually is if you go to figure 6, where they then use JAWS and optogenetics to target specifically this activity of the CRF neurons and see if specifically inhibiting that kind of time period of when the cells are active, they basically establish sufficiency that you can, you can just by doing that, you can have that behavioral impact that they show. I guess I was just thinking that to really get at kind of the specificity and importance of that activity during those time periods, it might be interesting to compare that to a protocol where you're either randomly inhibiting or specifically inhibiting at other time periods during the session to really see whether it's specifically that activity during those time periods that is kind of crucial here for the behavior.
I agree with you. I think another elegant experiment will be you just shine the laser during extinction, but the period without CS, and to see whether I think that can further prove whether the activity during CS will be less necessary. Yeah.
Yeah, and another thing along those lines, which gets more complicated, certainly, it's easy to come up with more complicated experiments, but if you compare to the activity, during the delay extinction, the activity kind of slowly over the course of the extinction session gets lower and lower. And so one could, during immediate extinction, to some degree, try to mimic that more biologically kind of real kind of situation. In particular, if one was using Invoke, where you could actually be monitoring the activity levels and sort of titrating your stimulation, you might actually be able to sort of kind of ramp, like gradually increase the amount of suppression over the course of the session to try to mimic more, what one sees during delay, during the delay extinction. And similarly, one might during delay extinction try to mimic more what's happening during immediate extinction in a more accurate and subtle way than sort of just turn off or completely suppress, essentially.
Yeah, that's an elegant experiment. Yeah.
Okay, I think we've covered all of the questions that have come in. So I think we can probably go ahead and wrap things up. Thanks to Kyesang for walking us through this interesting publication, and thanks to everyone else for attending and sending in questions. We hope this was scientifically stimulating for everyone, and that you'll join us again next week when we'll be going over another recent circuit neuroscience publication. We hope everyone will please stay safe and healthy, and hopefully we can all get back to our regular research activities soon. So take care, everyone, and thanks again, Kyesang.
Yeah, thank you.